Hello friends, welcome to BMH Learning. In this video, we are going to talk about another signaling mechanism known as RAS-MAP kinase pathway. Before we discuss the mechanism, let's know the different components which are involved till the activation of the RAS protein. So the first component we have here is the receptor tyrosine kinase itself. We already know about this, so there is no need to discuss about this. If you do not know, you can check the link in the description box. The second component we have here is the RAS. This is a G protein and we have already encountered this in GPCRs, but the G protein involved in GPCRs is trimeric protein, which is composed of alpha, beta and gamma subunits. But here in this RAS case, this is a simply monomeric G protein composed of only one subunit. Now, what is the meaning of this letter G here? This is representing guanine nucleotide. Okay, so this is guanine nucleotide. Now these G proteins, they have tendency to bind both GDP as well as GTP. Okay, now the binding of the GDP or the GTP is going to decide whether the G protein would be active or inactive. So if GDP is bound to RAS protein, this would be inactive. Okay, and if GTP is bound, this would be active. Now, unlike trimeric G proteins which are directly coupled to receptors, the RAS protein is not directly linked to receptor tyrosine kinase. This is very important. In case of the GPCRs or the G protein coupled receptors, the receptor is directly attached to trimeric G protein. But here in RTKs or receptor tyrosine kinases, the monomeric G protein or the RAS is not directly linked. So what does this mean? This means the activation of the RAS proteins is not that easy as in case of GPCRs, where following the ligand binding activation of the receptor, that activated receptor instantly stimulates the coupled trimeric G protein. So the question here is what to do now. To make this situation easy, the RAS proteins, they take help from another proteins which indirectly link these RAS proteins to receptor tyrosine kinases, and that protein is known as GRB2. So the third component we have is the GRB2 protein. This is a linker technically known as adapter protein which links the RAS protein to the receptor. Now this GRB2, it has two domains. First one, the SH2 domain, and the second we have is the SH3 domain, and we are going to discuss about these domains later. Finally, we want to know how GDP is exchanged with GTP on the RAS protein. So this is done by another protein which is called as SOS. So the fourth component we have is the SOS. Okay, so this is the guanine nucleotide exchange factor which binds to the inactive RAS bound with the GDP, and the binding of the SOS to the RAS protein exchanges the GDP for GTP. Now let's discuss about the mechanism. This is the diagram. First point we have is the following the hormone induced activation of the receptor tyrosine kinase, a complex is formed at the cytosolic phase of the plasma membrane, and this complex is made up of four parts: the receptor tyrosine kinase enzyme, the GRB2 linker, SOS, and the RAS protein. Now the formation of this complex depends on the dual binding ability of GRB2. Now what does this mean? Let's talk more about GRB2 structure. The GRB2 contains two types of domains, the SH2 and SH3. These one. Through its SH2 domains, it binds to the phosphotyrosine residues which are present on the cytosolic portion of the RTK. Okay, and by its SH3 domains it binds to the proline residues which are present on the SOS protein. So there is a distinction between SH2 and SH3 domains. The SH2 domains bind phosphorylated tyrosine residues, whereas SH3 domains, they bind to the proline residues. So this way it has dual binding ability because it binds both to the receptor as well as SOS protein through its SH2 and SH3 domains. Now let's know how GDP is exchanged for GTP. Following the ligand binding activation of the receptor, the SOS protein, which is the guanine nucleotide exchange factor, this comes very near to the RAS bound with GDP. Okay, the binding of the SOS to the RAS protein releases GDP. So what we are going to see, GDP is released and GTP is coming in place of GDP, because after the release of GDP the nucleotide binding site becomes empty. In that place GTP comes. Next we have is the activation of another protein which is known as RAF protein. Once the RAS protein is active by the binding of GTP, it acts on another protein which is known as RAF. Okay, let me use another color. That protein is known as RAF. This becomes activated by the action of RAS. Okay, RAF is a serine threonine kinase. Okay, now we want to know how it becomes active and who is responsible for the inactivation of this RAF protein initially. This RAF protein is actually the central point in regulation of this whole pathway. So let's know its structure first. This is the structure of the RAF protein. This is formed of two subunits. The first one we have is the N-terminal regulatory domain in pink color. Okay, and then we have the C-terminal kinase domain, this green. The RAS protein binds on the N-terminal regulatory domain. Initially, when the ligand is not bound, this protein, the RAF, is bound by another protein known as 14-3-3. This is a phosphoserine binding protein. That means this is only going to bind on the serine residues but in phosphorylated form. You have to remember this, that it only binds to the phosphorylated form of the serine. Okay, now this 14-3-3 is binding on two positions here. One is the serine residue which is present on the N-terminal regulatory domain, and another one which is present on the kinase domain. Now the binding of this 14-3-3 on the N-terminal regulatory domain is what keeping this RAF protein inactive. Okay, so when RAS protein comes and binds here on the RAF protein, it dephosphorylates this serine residue, and dephosphorylated serine residue is not the ideal substrate for 14-3-3. And what do we see? This 14-3-3 releases the N-terminal regulatory domain, and in this way the RAF becomes active by the action of RAS protein. So once this happens, you can see here the RAS is perfectly bound to the RAF protein, and this is in active form now. The next step we have is the activation of the MAP kinase by RAF protein. So we have here RAF protein in active form. Okay, this protein is going to phosphorylate one enzyme which is known as MAPK. Okay, this is here in yellow, and by the action of this RAF protein, this is phosphorylated and becomes active. In process, this enzyme is going to phosphorylate another enzyme which is the important one, the most important, MAP kinase. Here it is phosphorylated. So we have activated this enzyme, the MAP kinase, which after its activation goes into the nucleus and initiate the transcription of the genes.